Hey everyone, welcome back to another video by Camroom 5. This video is also going to be about the biology ATP examination where we will be designing experiments. So without wasting time, design an experiment based on method used by the student to investigate the effect of the enzyme. So there's some enzyme and its effect has to be checked on the production of juice from different varieties of apple. So here you are concerned with your different enzymes. Same enzymes and different varieties of apples. So your step 1 should be take 5 test tubes. You always see that I start my experimentation like this. Take 5 test tubes and say label A to E. Then you can mention take same volume and concentration of given enzyme in each. You can talk about use measuring cylinder for this. So you need to give an idea of the apparatus. So use measuring cylinder for this purpose. Now, how are these setups different? So you mention that take 5 different varieties of apple because your concern is apple varieties of apples, but apple can't be directly put into the test tube. So talk about make a paste using blender, then take same mass of paste from each variety because you can't change the mass, right? Varieties is already different now, so you can't change the mass anymore. So take different varieties, make their paste, but add the same mass of paste from each variety in each test tube. So apple variety A goes into test tube A, apple variety B goes into test tube B, apple variety C goes into test tube C, and vice versa. And then measure and then talk about what will you observe, what will you see. So you can talk about how much juice is produced here. They're concerned with the production of juice, right? So you're concerned about how much juice is produced. So you can talk about filter after same time, and then talk about filtration to collect the juice, and then talk about how you will compare. So you can say allow same time, and then say filter the test tubes to collect and measure juice volume. So here you were taking 5 test tubes, each had the same enzyme, same temperature, same time given. You are using even the same concentration. The only thing different is the varieties of apple were different, and even the apples weren't taken, their mass of paste was taken, and you have already talked about using a blender to make the paste. Then you talk about filtering out the juice after same time because you can't give more time to one and less time to the other. You have to give same time for a fair comparison. And then say, okay, we will measure and collect the volume of the juice. Now, name the testing solution the student used, state a temperature at which the test was carried out, and conclusion for the student's observation. Let me see, they're concerned about reducing sugar. I didn't read the whole question because I'm really quick right now. So testing solution is going to be Benedict solution. You can use Benedict solution. Benedict solution works best at 80 degrees Celsius. And if you see a color change of red, so you can say reducing sugar present. My recommendation is reading the question in detail, but I am a little quick here, so that is why I am not doing it. A student wanted to investigate how beetles respond to humidity. 10 similar beetles were collected from beneath leaves on the ground in a forest and taken to the laboratory. In the laboratory, the apparatus shown had been set up and left in place for 15 minutes. Okay, the stopper was taken out of the central hole. The beetles were put into the container and the stopper replaced. Suggest why the student waited for 15 minutes before introducing the beetles. That is obviously for conditions to become stable. For conditions to become stable. So that the heat and the light and everything is distributed equally. The dry part of the container had been treated with a chemical drying agent, such as one advantage and one disadvantage of doing this. So you can chemically make sure that the dry paper has no moisture on it. And disadvantage is that when you're using a chemical drying agent, it might, interfere with beetle movement. So might interfere with beetle movement because beetle might smell it. Beetle might like the smell, beetle might not like, beetle might not the smell might not appeal to the beetles. So maybe that's a problem. Advantage is to make sure all moisture is gone. All moisture is gone. Now we are concerned with part C only. After 20 minutes, the student observed that all beetles had settled on the moist paper. Design an investigation using similar apparatus to show the effect of light. So you're concerned with the effect of light. So what you can do, you can say, now you're concerned with beetles, right? So you can perform everything in the same apparatus because your beetles are going to be different already. So take an apparatus and at least 10 beetles in it. What you can do next is that you can make sure that conditions are different. How? So you can say allow a lamp on one extreme side of apparatus and make sure less light is available on the other extreme side. So you can create a contrast on the other extreme side. How will you do it? So mention that use a dark paper to cover the other side. This way you are making one side of the apparatus really brightly lit and the other side is pretty dark. Now what happens? Make sure the moisture, because you already know moisture is a very important factor, so make sure moisture, temperature and availability of food is same throughout. Because you want to see how light is affecting. So only light is different because one side is light, other side is dark, but everything else is the same. Now what will you observe? You will observe the movement of beetle for same time. Movement of beetle, let's say for 15 minutes using stopwatch. Now what are your expectations? You can say count number of beetles in the region brightly lit or lit and number of beetles in the region which was dark, and then compare. So here you're using the same apparatus, but this time the main comparison is one side is brightly lit, the other side is dark, and everything else is same, temperature, moisture, and now you're just allowing them to move, and after that you will count them. I hope you are getting a very good grip on how to answer such questions. In the next video we'll do 2 more. Stay tuned guys. Thanks.